Hello, I'm on the 2017 ASCO annual meeting, and I present here the updated efficacy data on daratumumab in combination with lenalidomide and dexamethasone, or bortezomib and dexamethasone in relapsed and refractory myeloma patients based on cytogenetic risk status. The Castor and Pollux trials did investigate the efficacy of daratumumab when added to standard of care treatments in relapsed multiple myeloma in patients with equal or more than one prior treatment line. And the primary efficacy analysis was already published. Here now we show for the first time the 1-year PFS updates after now extended median follow-up. So for the DRD regimen, the combination of daratumumab, lenalidomide and dexamethasone, the median PFS is still not reached compared to 17.5 months in the standard lenalidomide dexamethasone arm. And in the Castor trial for the DVD regimen, daratumumab in combination with bortezomib, dexamethasone, we have now reached the median PFS with 16.7 months. And this is also highly statistically significantly better than the standard arm VD with 7.1 months. So that also with this extended follow-up, daratumumab in combination with standard of care regimens significantly prolongs progression-free survival in this patient population. Based on cytogenetic risk status, we saw that the median PFS for high-risk patients for daratumumab, lenalidomide, dexamethasone is 22.2 months compared to 10.2 in the Lendex standard arm. This has a strong trend for significance due to the low patient number, and the standard risk PFS is still not reached for daratumumab Lendex. So also here highly significant benefit. In the daratumumab bortezomib dexamethasone combination, high-risk patients had a significantly superior PFS compared to the VD standard arm, as well as standard risk patients did. The cytogenetic risk was in this trial determined by NGS methodology for the first time. There was a concordance assessment with classical FISH analysis, which showed a high concordance rate between 88% and 100% of both methods. Regarding the secondary endpoint over response rate, after this extended follow-up, we see that the addition of daratumumab in both trials significantly improved over response, and more than that, it improved the rate of really deep responses, so the stringent CR and CR rate. All patients in the Pollux and Castor trials received minimal residual disease assessment in a pre-specified analysis, and the first data were already shown at the last ASH meeting. Here, the MRD risk status was correlated with the cytogenetic risk status, and we saw that high-risk patients only achieved minimal residual disease negativity in the daratumumab arms in both trials, whereas high-risk patients never received MRD negativity state in the standard arms. The MRD negativity rate was significantly higher in standard risk patients when daratumumab was added. Despite a very low number of MRD negative patients in the high-risk population in both the Castor and Pollux trial, those patients who were high-risk and MRD negative did not relapse during the follow-up period. So that we see in this updated analysis that the significant benefit in PFS is stable with daratumumab, and that we see that independent of the cytogenetic risk, the patients benefit regarding PFS over response rate and MRD negativity, and the survival data are awaited later this year. The combination treatment, daratumumab, lenalidomide, dexamethasone, and daratumumab, bortezomib, dexamethasone, was recently approved by the EMA and is now also in the daily clinical reality for our patients.